protein synthesis. Protein synthesis starts in the nucleus. We have, this is our nuclear envelope, DNA. So there's a really, really long, big, massive molecule of DNA in the nucleus. We unravel it in the region of a gene, so it becomes unwound. So that's due to the enzyme helicase. What happens then is you get free mRNA nucleotides lining up alongside one of the strands. Just one of the strands is acting as a template, so this is different to semi-conservative replication. Also, the enzyme involved is RNA polymerase, because you're going to make an RNA molecule from a copy of the DNA. So the DNA molecule unwinds, then the hydrogen bonds break, and the base sequence is revealed, and the free RNA nucleotides line up by complementary base pairing. Remember, A and U this time, because it's U's in the mRNA, and you've got T in the DNA will line up with A, G and C and C and U. So this is our complementary base pairing of the mRNA. Then the RNA polymerase seals the sugar phosphate backbone by forming the phosphodiester bonds. This is called pre-mRNA because it needs to undergo splicing, which happens in the nucleus, and effectively there are introns and exons, and you remove the introns, so those are removed using enzymes, and you end up with mRNA, which is just exons. The mRNA then leaves the nucleus through the nuclear pore. When it gets into the cytoplasm, it associates with a ribosome. The ribosome is able to pick out the first codon. The codon on the mRNA will have a base sequence, and that base sequence, for example AUG, will be complementary to the anticodon of the tRNA. The tRNA will be attached to a specific amino acid that will correspond to the anticodon that it has. So each anticodon will be specific to one particular type of amino acid. Now, because of the nature of the code, there's actually 20 amino acids, but about 64 different codes. So we say that the code for amino acids is degenerate. That means that if you have CCU and CCG, they might both code for the same amino acid, isoleucine, for example. And you might have lots of, you might have even 4, so you could even have C, all coding for isoleucine. So it depends on the amino acid, but we say it's degenerate because the first 2 must be the same, but the last one can be slightly different, and you still get the same amino acid being coded for. So in this case, again, it's complementary base pairing. The ribosome holds over the codon, the anticodon will come in and form hydrogen bonds. They must be complementary, so if it's A on the mRNA, it'll be U on the anticodon, U on the mRNA, A on the anticodon, G on the codon, C on the anticodon. Then the ribosome will move along and it's able to highlight 2 codons at the same time, and that allows 2 tRNAs to come close enough together to allow a peptide bond to form between the amino acids. As the ribosome moves along, that allows the bonds between the complementary bases to break, and so that tRNA will be removed. So that tRNA goes back into the cytoplasm to pick up a new amino acid. So notice the amino acid is gone. That amino acid is now joined to this amino acid, and the process repeats. And the ribosome tracks along the mRNA, highlighting another codon, and it moves along and along and along till it gets to the end. When it gets to the end, the final codon does not match any anticodon on any tRNA, and as a result of that, everything detaches and you end up with your polypeptide sequence of amino acids, which is the primary structure of your protein. Once you've made that with your ribosomes and your rough endoplasmic reticulum, it may then be transported in a vesicle to the Golgi for modification with carbohydrates. So it's important that this base sequence stays the same, because if the base sequence is different, then that could change the amino acid that you code for. If it changes the amino acid that you code for in the primary structure, then you could end up with a change in the secondary structure and where the hydrogen bonds form, and that will affect your alpha helix formation, and your beta-pleated sheet, where they form, and then that will affect your tertiary structure, which is the further folding, and that's held together by your hydrogen bonds, disulfide bridges, ionic bonds, which are basically between R groups that have opposite charges. So if you change the amino acids that are in that structure, then you will change where the folding is. If you change the folding, you change the final 3D shape of the tertiary structure, and that will mean if it's an enzyme, then the active site may change shape, it won't be complementary to the substrate won't fit, you won't get a reaction. If it's a receptor, the substrate won't bind to the receptor. If it's an ion channel, the ion won't be able to move through the ion channel. So it's very important that the primary structure doesn't change, so that the secondary structure and the tertiary structure and the final 3D shape of the tertiary structure stay the same, the charges stay the same, and the shape stays the same, so that you don't end up having a non-functional protein.